My name is Alex Carnes, and this is Carbohydrates of Physiological Significance. So, there is a large biomedical importance of carbohydrates. The liver acts as a metabolic hub by converting most carbohydrates into glucose, which can then be used for energy or stored for later use. Glucose serves as the body's primary fuel, providing energy for most cells, um, and being especially critical for the brain and red blood cells. Glucose also serves as a biomedical precursor, providing the building blocks needed to produce glycogen for storage, as well as nucleic acids and glycoproteins for cellular structure and function. Disruptions in carbohydrate metabolism can lead to different diseases such as diabetes, which affects blood glucose regulation, and also lactose intolerance, which impairs the digestion of certain sugars. So, in this article that I found, which was called The Brain Glucose and Ketone Utilization in Brain Aging and Neurodegenerative Diseases, it had this really cool fact on the brain, and it said that the brain only represents about 2% of the total body mass, yet it can spend about 20 to about 23% of the total body energy, corresponding to about 110 to 140 grams of glucose per day. So this is a cool photo here, and it shows how the liver and muscle both use glycogen. So in the liver, you have glycogen, and then it is converted into glucose 6 phosphate, and then converted to glucose, which then regulates the blood glucose levels. Whereas in muscle, you have glycogen, which is converted to glucose 6 phosphate, which then is used for energy and movement of that muscle. So, what are carbohydrates? Like I mentioned before, carbohydrates are organic biomolecules that primarily function um, as a major source of energy. They are made of carbon, hydrogen, and oxygen. Carbohydrates are polyhydroxy compounds, meaning that they contain multiple OH groups that make them highly soluble in water. They can be classified as aldoses or ketoses, depending on the aldehyde or ketone functional group that they have. Functional groups in carbohydrates, like three that I just mentioned, the aldehydes, ketones, and hydroxyl groups, um, they control how the molecule reacts chemically by influencing properties like oxidation, bonding, and participation in metabolic reactions. So, small differences in carbohydrate structures can significantly um, change how they are recognized and produced and processed by enzymes, which in turn affect how they are metabolized and what roles they play in the body. So, this image here, uh, it has the ketose and then the aldose, which you can see they both have these polyhydroxyl groups, which are the OHs. And then in the ketone group, you just have a ketone. So it's a carbonyl carbon with a carbon attached to another carbon. And then the aldehyde is just the carbonyl carbon attached to a carbon and also attached to a hydrogen. So there's a couple types of carbohydrates here. We have the monosaccharides, which are just the simplest carbohydrates. They consist of single sugar units, such as glucose, that can be quickly used by cells for immediate energy. And then you also have disaccharides, which are made of two monosaccharides that are linked together and must be broken down by enzymes during digestion before they can be absorbed and used for energy. And then the final one here is the polysaccharide. And polysaccharides are just long chains of monosaccharides um, that function mainly in energy storage, like in glycogen, or also as structural support, like in cellulose. Um, from another article that I found online called Blood Groups and Red Cell Antigens, oligosaccharides um, are pretty much just short chains of about 3 to 10 monosaccharides, and they act as markers on the surface of red blood cells. Uh, they can help determine a person's blood type in the ABO system, which specific sugar chains patterns form A, B, or A1 antigens. So, glucose, like I've said before, is the central molecule in metabolism, which serves as the body's primary fuel and maintaining stable blood glucose levels to ensure that a continuous energy supply for cells, especially in the brain and in the red blood cells. So, glucose acts as a universal metabolism intermediate because many other dietary sugars are converted into glucose or glucose derivatives or glucose derived intermediates so they can enter the central energy producing pathways. Glucose produces energy um, and building materials by being broken down to produce ATP while also supplying intermediates needed for biosynthesis and other important biomolecules. In aqueous solutions, glucose predominantly exists in a stable cyclic ring rather than the linear chain, which can affect its chemical reactivity and biological function. So this is a cool little photo of different glucose levels. So you have your hypoglycemia, which is the low sugar. You have your normal levels, and then you have your hyperglycemia, which is the high sugars. Um, and these really show um, how they correspond to how tightly blood sugar is regulated in the body. Um, the Mayo Clinic has an article and it explains that in a fasted state, glucose is normally maintained in the range of about 100 milligrams of glucose per deciliter of blood. And it explained that in hypoglycemia, that level was significantly lower, and that in hyperglycemia, uh, that it reflects a more chronically elevated glucose levels above 100 milligrams per deciliter. And then when regulation is impaired, you can develop diabetes, which I'm sure many people or many of you guys have heard. And that is pretty much a blood sugar level of greater than 126 milligrams per deciliter. Glucose structure. So it comes in many different forms, but um, glucose can, in fact, exist in a linear form, but in aqueous solution, like I mentioned before, it is predominantly converted into a cyclic ring structure through an intramolecular hemiacetal reaction between the carbonyl and hydroxy group. In fact, about 99% of glucose that is in ring form in solution. The most stable form of glucose is the pyranose structure or pyranose ring, which is a six-membered ring that is favored because it has reduced steric strain. Glucose in the pyranose ring most often adopts a chair conformation, which minimalizes steric strain, like I mentioned, between atoms and places substituents in the most stable positions, giving it the lowest energy and highest stability form. So the anomeric carbon is very important. It is a special carbon in the ring of glucose where the shape can differ. Um, and it creates either the alpha or beta form, depending on which direction a specific OH is pointing. So this is my chair conformation you can see here. And then the anomeric carbon would be this carbon down in the bottom right, which is carbon number 1. And since the OH group is pointing up or in equatorial position, this would mean that this is a beta. And so this chair conformation is showing the beta D glucose. Isomerism. So um, this is pretty much just when a molecule has, the same um, molecular formula, um, but can be drawn in different ways that are different from each other. So this is kind of how in these different forms here, uh, they are applicable and different in real life. So in the D versus the L, enzymes in the body mainly recognize D sugars, which are biologically active forms used in metabolism. Um, and then, like I mentioned on the last slide, between the alpha and beta, the positions of the OH group on the anomeric carbon differentiate between the alpha and beta forms, and it can change how the sugars behave and is used in biological reactions. Epimers are sugars that differ in just one carbon atom, such as the glucose and galactose, like I've shown, which can still lead to very different biological roles. So as you can see here, in the galactose, your carbon on the left here, its OH group is pointing up and the hydrogen is going down, whereas in the glucose, it is flipped and the OH group is going down and the H is going up. So that's what make these two epimers. Uh, and then like I said in the other slides, whether the sugar has an aldehyde or ketone group affects how it reacts and how it enters metabolic pathways. So a glycosidic bond forms when two monosaccharides join together to make a disaccharide through a condensation reaction where a water molecule is released as a bond is created. Alpha and beta linkages describe how the sugars are connected, and this difference affects both the shape of the molecule and whether humans can digest it. For example, alpha linkages are generally digestible, while the beta linkages are often structurally harder and could even be impossible for us to break down. Um, then we have enzymes, which are highly specific. Each type of glycosidic bond requires a particular enzyme to break it, meaning different sugar linkages are digested by different enzymes. And then we have common disaccharides, which include sucrose, lactose, and maltose, each made by two monosaccharides linked by glycosidic bonds and serving different roles in diet and metabolism. This is a really cool uh, graph here, and it shows the prevalence of lactose intolerance in recent populations. Uh, and so, first of all, what makes people lactose intolerant is that they don't produce enough lactase, which is the enzyme that breaks down lactose. And as you can see here, that um, this is genetically inherited. So places like South America, Africa, uh, and then in Asia, they have a high prevalence of lactose intolerance, whereas in Europe, especially Northern Europe, they have a very low prevalence of lactose intolerance. So we have here your polysaccharides. Starch, for an example, is a storage of polysaccharides made of amylose and amylopectin that plants use to store glucose for energy. Glycogen is a highly branched storage form of glucose that allows animals to quickly release energy when needed. And then cellulose is a structural polysaccharide in plant cells with a beta 1,4 glycosidic bond, making it rigid and indigestible to humans. So the structure really does matter, as the amount of branching affects how easily enzymes can break down the polysaccharide. So with more branching allows faster glucose release. Glycemic index is a measure of how quickly a carbohydrate raises blood glucose levels after it is eaten. A simple carbohydrate will have a high glycemic index because they are absorbed faster, causing rapid spikes in blood sugar. Complex carbohydrates will have a low glycemic index because they're digested more slowly and lead to a gradual rise in blood sugar. Glycocalyx is a carbohydrate-rich layer on the surface of cells that help protect and coat cell membranes. So the glycocalyx is important for cell signaling because it allows cells to identify each other and support immune system responses. So in this little model here, you can see that the glycocalyx is made up of these red carbohydrate chains, and they are attached to membrane proteins and the lipids on the outside of the cell membrane. So this is very important. These are my multiple choice questions. So, which statement best explains why glycogen is more effective than starch for energy storage in animals? A, it contains beta 1,4 linkages. 2, it is less soluble in water. Or that was B. C, it is more highly branched, allowing rapid glucose release. Or D, it is composed of different monosaccharides. 2, carbohydrates are chemically defined as hydrocarbons with nitrogen groups, hydroxyaldehydes or ketones, C, lipid derivatives with phosphate groups, or D, amino acid polymers. 3, which structural feature of glucose is responsible for the difference between alpha and beta forms? A, the position of the carbonyl group. B, the number of hydroxyl groups. C, the orientation of the hydroxyl group at the anomeric carbon, or D, the length of the carbon chain. 4, which type of carbohydrate is primarily involved in cell recognition and can contribute to blood type determination? A, monosaccharides. B, disaccharides. C, oligosaccharides. Or D, polysaccharides. 5, which polysaccharide is highly branched and serves as the main storage form of glucose in animals? A, glycogen. B, starch. C, cellulose. Or D, chitin. Here are the answers for those questions. And then this is my works cited, and then my images cited with the URLs.